Hello everyone, welcome to, what month are we in? October. October's talk. You blink and it's next October. Welcome everyone, thanks for turning out. Yeah, I've got a very exciting talk tonight, but as usual going to start off with the background of everything. First of all, thanks to Thirsty Giraffe for setting all of this up, massive help. Thanks to Painted Dog TV for filming this. If you guys know anyone that wants to watch this afterwards, please just visit Painted Dog's YouTube channel and they can watch it. And then finally Scales. Scales definitely help us out with quite a lot of behind the scenes stuff, so massive thank you to them. And then finally thank you to the Nature on Tap team who are hiding all over the show here. And thanks to all of you. Thanks for everyone for coming out. It's always a great turnout. It makes us very happy. And we've got a great talk tonight. Dr. Janine, she asked me not to call her doctor, but it's Dr. Janine, is going to tell us all about her work with Rhino Force. She's the veterinarian for Rhino Force, and I'm not going to waste any more of your time. We're going to get straight into it, and I'm going to hand over.
Thanks, guys. Dr. Kyle. Hi everyone, let me just check if this works. Okay, thank you all for coming and joining my talk tonight. Yeah, I'm going to talk about assisted reproduction in wildlife. I'm working with Hemmersbach Rhino Force in that field to set up a biobank actually, and also to do research. Before I go into all the details about what assisted reproduction is and why it's important, I want to just quickly introduce myself and the company I work for. So, as you can see, I always had a passion for animals, especially wildlife, and wanted to be a vet as many little girls, and I actually pursued the dream, became a vet, worked for a couple of years overseas in general practice and in wildlife rehabilitation in Germany, the UK and the US, before I set the exams again in South Africa to become a registered veterinarian in South Africa. And after a year of community service, some of you might know, but vets have to do a year of community service, I started working for Rhino Force. So Rhino Force was started by Hemmersbach, which is a global IT company, and they have the idea to give 20% of their profit to good causes through direct action. So they actually start a project and run it themselves, and one of the projects is Rhino Force. The people here who know Rhino Force will most likely think about the anti-poaching side of it, which is one of the sort of 3 projects that Rhino Force does. So the anti-poaching entails elite units that support the area, the Greater Kruger area, with anti-poaching, with new technologies and rangers. And then another project is actually community work in Massengier, where a lot of the poachers in the Kruger and Greater Kruger area are coming from. And the idea is to offer them a way out of the poaching and an alternative. So this is actually, while the anti-poaching is more of a symptomatic treatment in vet terms, this is kind of trying to target the cause of the whole problem. And then the third project is the Cryovault, which is a biobank for gametes, so mainly actually for semen, and other biomaterials, viable biomaterials that we in the future can use to increase genetic diversity, but I will go into that. As part of the whole setting up of the biobank and running it, we also focus on research to actually advance and research into assisted reproduction and how to implement them in wildlife. So this is not really one of the major focuses, but I wanted to mention it briefly. Another thing that Rhino Force does is offer support during crime scene investigations with the police. So when the police does an investigation, they obviously look at it from to collect evidence, ballistics, and only a veterinarian is actually allowed to say what the cause of death was. So if I'm there, then I can say this animal died 100% of poaching for this and this reason. I document it, report it, and if it goes to court, then it has a bigger chance of standing up in court. So to go back to why the Cryovault and the biobank was started, to give a bit of a background, most of you who are in conservation or interested in conservation will know that we are facing, or that we're currently at the 6th mass extinction, and we're facing the loss of genetic diversity, of the loss of animals on a greater scale than is actually normal. Most of it is due to the human population growth and everything that comes with it, from resources that humans take, to habitat destruction, fragmentation, and all sorts of things. And we mainly focus on the rhino, but the assisted reproduction is also applicable to other species. But most of the research that we do is actually focused on rhino. And I've already mentioned the causes of death for all sorts of species, from habitat fragmentation and destruction. The rhino faces an additional threat, and most of you know, but it's the ongoing poaching crisis and the killing for horn and for profit. So this is why this is what the consequence of this already is. So the black rhino as a species is critically endangered. 2 of the subspecies are also critically endangered, and then the white rhino is only actually considered near threatened, despite the declining numbers in the wild. But if you look at a subspecies, the northern white rhino, the northern white rhino is actually functionally extinct. So with only 2 remaining females, no male left, there is no way that the species can rebreed itself. And again, this is where assisted reproduction is the only thing that can save it, although it might already be too late. And some of you might have heard the headlines. There was a breakthrough with embryo transfer, and unfortunately the mother died of unrelated causes, and it's assumed that the fetus that they found was actually one that survived embryo transfer. So I just wanted to put this in here because this is just, like, people are looking at bringing the mammals back, which, okay, it's already extinct, but this is the possibility of applying those advanced techniques. That's what you could do. So for the northern white rhino, you can actually make an embryo and put it into a southern white rhino that then gives birth to northern white rhino. Again, I will explain that a bit more in detail. So for the rhinos, the conventional conservation efforts consist mostly of habitat protection, anti-poaching to counteract the poaching, and then to allow natural breeding. So population management, making sure that they can actually have offspring in the wild and get that offspring to reproduce in the future. And then obviously also community work has become a bigger role, because that's where the whole problem starts. And as part of all this, intensive population management has become necessary. And you all know dehorning, ear notching, collaring, translocations, all of those are something that happens on a daily basis. And they all require immobilizations. So they offer actually an access to animals that we want to use for to do research and also to collect viable material. So this might look like a bit of a difficult slide, but that's to explain what assisted reproduction is, and then also where it can play a role and how it works. So you see, you have a male rhino and you have a female. So when they have natural breeding, they have a calf that's 50% mother, 50% father, genetically. Then if you collect that semen from that male and you freeze it or you transfer it directly to do an artificial insemination of another female, you again get a calf that's 50% this female and then 50% of a male. That doesn't necessarily have to be in the same area. And if you go even a step further and you collect eggs from a female, you do ICSI, I explained that just now, but you basically make a little embryo out of the body. You get that embryo and you transfer it into an unrelated female, then you actually get a calf that's not related to that surrogate mother.
Sorry, there's just kids here at the back.
Oh, is it?
Let's see if I can do that.
Don't tamper with it too much, it's temperamental.
There we go.
Cool. So then you get a calf that's not even related to that female, to that surrogate mother. So that's where it becomes quite interesting. The potential of it is that if you preserve you can preserve the genetics of all those animals, but you can introduce the genes or the embryos into isolated or fragmented populations. So bring in an animal without actually moving an animal. And for everyone who knows what a rhino translocation entails, that's quite an important step. So when you translocate an animal, you're limited to that one animal. You're limited in distance because you have to consider the animal factor, the stress, how they will do after the translocation. Also, that often they don't adapt very well if it's an environment that's that they're not used to, where there's plants that they don't like, or diseases like trypanosoma. All those things have to be taken into consideration, and then also it's only limited to the genetic of that one animal. So if you move a male, he has to become dominant to breed, and then in that area he will probably be the dominant breeding male. So then the next generation are all half siblings. So you're limited with that. While if you would bring embryos in, or at least semen from several males, then you have a whole tool to quicker introduce new genetics. And the other thing is that you can prolong the reproductive lifespan of an individual beyond its natural life. So that means death, or the reproductive life in the way of that animal might be too old, or it might be injured, or anything like that. Okay, back to the biobank. So I've tried to sort of make a way of explaining what actually happens. So we can collect the gametes, which is the semen and the eggs. Then we cryopreserve them. Eggs actually don't freeze very well, so it's better to try to make embryos and freeze them. And then create a genetic reservoir that can sit there when it's maintained in liquid nitrogen forever. So it's there is no time frame in it. So we can freeze something today, and 10 generations from now they could use us, as long as we maintain the levels and as long as we maintain the integrity. And then in the future we can actually try to regrow viable populations and add to species survival. Sampling opportunities that we as the Cryovault look at is post-mortem and poached and euthanized rhinos. Obviously, poached always comes with a time limit. If we have a hot day, or if an animal is laying out in the sun, then we're very limited. Then we're talking about hours, but under ideal conditions we can collect up to 24 hours. And then live during all those management interventions that would require immobilizations anyway. As I listed before, dehoning, ear notching, collaring, translocations. To do those collections, live and post-mortem, we have to have a good understanding of the anatomy of the animals. And in wildlife, not much is known. So we actually had to go ourselves first and also fill in the unknowns, fill in the gaps. And I've actually made these drawings to sort of, give a better understanding of the anatomy. And these are examples of post-mortem collections. So this was from a poached rhino. That's actually the testis, and then we take the epididymis off. So that's the storage area of the sperm. We inject it with an extender, and then collect the semen in the tube. And if of good quality, we can still freeze it. And then this is an ovary of a rhino that I don't extract the eggs myself, so I actually package them and then send them to Onderstpoort to the IVF lab, and then they do the extractions and, if viable, go to the next steps. So we face quite a few challenges. Like I said, those management interventions are good opportunities. but we sit with a few obstacles to make it work. So time is a very important one. For those of you who have joined dehornings or any management interventions, most of the time we try to work as soon or as early as possible in the mornings, so to prevent to work at high temperatures. And then you have everything organized. There's a veterinary team, there's a helicopter, and as many animals as possible should be done that day. So if we come in and delay the whole thing, then it's not going to work. So we worked on that. Then the environment, if you think about a captive setting, if you have animals in zoo conditions, these animals, they can be ideally, they can be in ideal conditions. There is ventilation, they can be put on cushions, they get monitored, they can be intubated, there is like a whole team available, everything is under control. We work completely out of control. We don't have fancy equipment. Everything that we have has to be able to withstand wind, rain, sun, and also has to be light enough to be carried and has to be portable in the first place. And then thinking about a captive setting recumbency, so that means like the position that the animal is in. Most of these techniques that have been used in rhinos and were described in captive animals are actually done in animals in lateral recumbency, so on their side. When you join a dehorning, you will see most of the time the animal is kept in a sternal recumbency, so meaning like half on their chest. And that is to actually allow better airflow, because actually better ventilation, I should say, because the drugs used to immobilize the rhinos actually cause respiratory depression, and that's why we had to work under, in sternal recumbency and sort of again try to adapt to that. Like I said, there's limited data on wildlife in general, so there's not much that we can work with in the first place. And then we had another obstacle, which is the immobilization protocol. I'm not going to go into too much detail, but so free-ranging rhinos get immobilized with a potent opioid and then with a tranquilizer or sedative added. The normal protocol, the commonly used protocol is etophine with azapron, and azapron is a drug that unfortunately works against us and inhibits semen emission. So semen emission is when the semen actually leaves the storage area in the epididymis and gets pushed into the tubes. So we got very little samples or very bad sample quality with that protocol. So we had to look for an alternative, and medetomidine is an alternative. We use it with midazolam just for some extra effects. I don't want to go into the details of that, but that actually helps us in cats and dogs, for example. If they get immobilized with medetomidine or sedated with medetomidine, you can actually collect semen without anything else. You can just put a catheter in. But again, I'm going to go into more details. So because we had a lot of we were facing a lot of negativity or hesitation towards changing the protocol, and I've tried a few times with the azapron protocol, but it's like the success rate is very low. So then we researched this alternative protocol in black and in white rhino. We also published it, and we actually see that from the blood gases and the blood parameters, that there isn't much of a difference between what's been published for the azapron protocol and what's been published or what we found. Unfortunately, we still can't convince everyone. So now to the actual semen collection. So again, we have the drawing here of the male rhino, and when we do semen collection, we want to identify the area of the prostate, which I highlighted or circled here. And to do that, we use ultrasound. You can see we always do it with a smiling face. Then we have to clean the rectum first, I should say. So that is, I spared you the pictures of that. And then normally, in most wildlife, you have to use electroejaculation. Like I mentioned before, there's other methods, but electroejaculation is a method that was originally developed for humans with spinal trauma. So actually to get the ejaculatory reflex going without having to have a conscious person or person that can contribute as such. And as you can see, or as you can imagine, there is no such thing as an electroejaculator for rhinos. So we had to get creative and develop one. Oh, well, that's the probe. So the probe is the part that goes into the rectum and gets placed on top of the prostate. Let me show you quickly. Here. Then we have this little battery-powered electroejaculator that gets attached to the probe. Once the probe is placed, you see that it has three electrodes that get placed on top of the prostate. We use the prostate only as a reference area for the nerves that actually initiate the ejaculatory reflex. And then we need 3 people, so the one that does the stimulations. So we apply low voltage stimuli. So very, like, if you feel it, it's just like a tickle, and that just stimulates those nerves. And then we have to apply sets, rest, and then sets again of stimulations on and off to just get that reflex going. So the first person does the electroejaculator, then the other person holds the probe in place, and then the third person will collect the semen. Again, we've become creative here. This is actually a pool noodle that we cut, and then we put as an insulation, and we put a tube in, and it's a rectal glove that we cut the bottom off, so we can use it as a sleeve to cover everything that needs to be covered and collect the sample, hopefully safely. Yeah, I showed you already how this works. And so that's just, people always get scared by the size. Look at the size of a fecal ball of a rhino. What goes out goes in. So it's just the same sort of thing. It was taking too long though to get the whole ejaculation going. Most of the time we needed like 40 minutes, especially if it's not a breeding bull. And that obviously exceeds the time for any dehorning or any management intervention. And again, we had a lot of hesitation towards letting us try. And then I thought, okay, with the medetomidine in dogs and in cats, we can just put a catheter up the urethra, and because the semen sits there before it comes out, before it gets actually, what's the word, expelled, I think. And then I thought about, okay, maybe we can do the same in rhinos, but again, there is no such thing as a urinary catheter for a rhino. So we actually use a kid's feeding tube, which is still smaller than you think, but it works perfectly, it's long enough. And we just put it all the way up to the area of the prostate. This is what it looks like in the field. It's very non-invasive, but we always do the stimulations first. So we apply one set of stimulations, give it a bit of rest, do another set, and if there's good stimulations, then we keep it at the 2. If necessary, do a third set, and then most of the time it really takes only 5 to 10 minutes, and then we can put the catheter in. Here's a bit more in detail. For those who have never seen a rhino penis from close, it is very funny looking. And then we just put a syringe or our finger top on. Once the semen runs in with capillary forces, take it out and secure it in a little tube. These pictures are just from the field to show that we're actually working while dehorning. So I've had it this year that I was actually finished with the semen collection before the dehorning was finished. Just some more pictures from the field. This is on Salati, by the way, all of it. Thanks, Maddie. These are just some funny pictures because you also have to take it with a little bit of humor if you have to work and talk about penises and similar. And yeah, it requires a lot of effort sometimes. Not everyone is a giver. Not everyone is. Well, this one was a bit shy. So once we have a sample, we do some evaluations to see if it's actually feasible. Initially we set our values for all of these parameters that we look at 50%, but we've gone a bit away from that because with ICSI, as I will explain later, you only need one viable sperm, and if it's a really valuable bull, we will freeze it no matter what, or there has to be some level of quality, but we will adapt our our values. So this is just an example. it never looks like this anymore. With electroejaculation, because you stimulate the natural ejaculate, you get all the pre and after fluids, and you get like a lot of volume. Now with the urethral catheterization, we actually talk about 0.5 milliliter to 2 milliliters. And then we look at if the sperm swims, first of all. Does it swim straight? We give a sort of a percentage, which is a bit subjective. And then we look at the concentration, at any abnormalities, and the life-death ratio. There's certain smears and evaluations that we do. And in the field, actually, let me quickly go here. In the field, we just have a first evaluation to see, okay, do we have to rush to the lab and freeze it, or can we continue with the next animal in the field? So I'll go back to this. Don't know if I can, it's a video. I don't know if it, is it playing? Okay, yeah. You saw them swim, eh? Okay. So yeah, back on this photo. Okay. Back to this photo. If we decide we're going to keep the sample, we're going to process it, then we start already with some of the processing in the car, depending on how far we have to drive. If we have to drive 5 minutes, we don't have to extend it. If it's 10 minutes, 15 minutes or longer, then we put an extender with the sperm to make sure that it actually survives the whole process, and we keep it away from the heat. So we have a cooler box that's that kind of is adjusted to temperature. And depending on the day, either with heating pads or cooler pads. And then we go to the field laboratory. These are 2 examples. We take all our equipment with and set it up there. So this was just the guy's picket. It's like an outside counter, and we just set up there and actually did the freezing there. So for the freezing, again, there's like in captive animals, there are very, very difficult procedures, and that require a lot of equipment and step-by-step dilutions and all that stuff described that have the optimal quality to keep the sperm alive during freezing. We had to make it work with a very simplified protocol, because it has to work in the field, and we also want to kind of limit it in time. So we added cryo extender, not like it's most of the time done step by step, but we found a cryo extender that we can just add all at once. So a cryo extender has a cryoprotectant in it that sort of dehydrates the semen to some extent, or the sperm to some extent, that it actually doesn't burst during freezing. So it helps to prevent ice crystal forming and cryo damage. Then once that's done, we immediately load it into straws and seal those straws, and then we wrap them into a towel, and we put them in the fridge for about 30 minutes. After 30 minutes, we take them out, we put them on this little rack, and we freeze them over the liquid nitrogen vapor. So the liquid nitrogen has 196 degrees. And the vapor reaches about minus 80 degrees. So if we put this little rack on there, we get the gradual freezing. It goes still very quickly. After 15 minutes, it's done. And then we can just plunge them into the liquid nitrogen and transfer them into the storage containers, and then into this flask. Yeah, this is the same area there in the field where we just did the freezing. In captive animals, there's all sorts of directional freezers that do the process perfectly, but this has actually worked, and we had good quality. And just to give you an idea, so far what we've done, we've attempted semen collections in 100 rhinos, white rhinos, and 9 black, whereof 2 of the eastern subspecies and 2 of the southern central subspecies. And then we collected semen-rich fractions in 77 animals. Initially, when we just did electroejaculation, it was only about 50% or so, often even less, where we got a semen sample, and now we go to almost 100% semen samples. That doesn't mean freezable semen samples. As you see, feasible is just under 50%, but I think that's gonna, well, in our last attempts it was always more towards the 70%. Just because we've gotten better with our techniques, we know where to put the probe, and we've gotten quicker, and yeah, there's a lot of errors that we could eliminate. So then we also collect additional data, because there's no value in having just a biobank full of semen samples that have no background data with them. So we collect of every animal where possible a microchip number, some of the environmental data, where they were from, if they were breeding bulls, who they're potentially related to, what area they come from, so that we can in the future, if we use them, that we can use them wisely. And then we also collect a set of blood samples for disease screening and for DNA analysis. So if we ever want to use them, if you ever want to use the semen samples and, for example, import or export them, then we can actually show that they were free of diseases. We don't know yet what diseases will be found in the future, so it's always good to have a backup. And we also freeze tissue samples. In the future, there might be techniques that can use them to actually transfer the DNA into either sperm or egg. And there's just some pictures here to show what it looks like, what our freezer looks like. And so we want to just generate a whole set of data that accompanies each sample that we freeze, and also possibly use some of these samples for research. I'm thinking now about hormone analysis or things like that. And yeah, like I already said, if they ever get used in the future, that we can actually prove that they're usable. So that's on the semen collection in the rhino, but we've also worked in other species. That's not our major focus. But as you can see, there's some examples here, and I've just started to attempt semen collection in pangolins with Debbie. Where is she? There. We got our first sample. Unfortunately, most were dead, so we have to again look into how to improve the techniques. And I do want to look into wild dogs. So that's the male side. Now we go to this female side, which is a bit more complicated. So to get eggs, it sounds easier than it actually is. You can see again there's an anatomical drawing. And we have to get to the ovaries and actually empty the follicles. So the ovaries in a rhino are very far forward in the body. In a human, in a horse, the horns of the uterus, which are these, curl around and actually make the ovaries accessible transvaginally. So if you want to do ovum pickup in a male or in a human, you can do it transvaginally, while in rhinos they're so far forward that we cannot reach them. You can see that's the vagina here, so there's no way we get from there to there. So it's about 120 to 150 inside the body. So we have to, until there, we have to empty the rectum, which again is a lot of effort, as you can see with tubes, and we have a pressure pump with a generator and empty the rectum. And this is up here, this is the sort of the part that we aspirate with. You see the ultrasound here. And then you see a needle, and we can manipulate the needle from the outside. So we position the ultrasound against the ovary at the right angle through the rectum against the follicle, and then from the outside we manipulate the needle to actually go into the follicle. So here you see we have to do ultrasound first, of course, to know what we work with. Do we have an animal that's pregnant? Then we obviously stay away. What time of the cycle is she at, and how many follicles are there? What can we expect? We can stimulate rhinos, but not to the extent that it's possible in humans. Just check. Yeah, this is. These are some pictures from the field. So you see the external part of that, we call it OPU arm, so from ovum pickup arm, that we manipulate from the outside. We look on the screen, and then you see me, we're manipulating the needle. Just, how did I do this earlier? So there you see 2 follicles, and then you see the biopsy line, and you see the needle coming in. When you puncture the follicle, it always wants to push away, so you have to give it a little, nice little prick, then go back a bit, and then you see we're now aspirating and flushing it, and aspirating and flushing it. And we use a special media that it's a double lumen needle that we insert, and through one lumen we insert the fluid, to the other one we aspirate the fluid with the potential egg in it. And then we have everything pre-warmed, and there are containers where they get rescued in. And then, because we sometimes have a lot of fluid, we have to filter it first and capture everything that's potential egg. And then look under the microscope. So here you see there's one of our little dishes where the eggs are in, and you see one of the eggs here. In this, we call it the cumulus cell. And then you see all the ones that we harvested from that one female. They're here there's one there. And then we package them safely, securely, and then they get actually packaged in a big box, and we try to maintain temperatures between 18 and 26 degrees during the transport to the IVF lab, which can sometimes be challenging. So the farm where the private rhino breeder where we've been doing this is in the Northern Cape, and the eggs have to go to Onderstepoort. So the bateleurs were actually good enough to help us there, because before we tried it with the public way to drive to Kimberley, fly to Johannesburg, then drive to Onderstepoort, and there's obviously a lot of time delays and also a lot of times when it can go wrong temperature-wise. So the 2-hour transport with the bateleurs is obviously a lot better. Then Mario, that's the guy who runs the IVF lab at Onderstepoort at the Veterinary Faculty. He gets the eggs and he looks for them. And so when you think about any animal, including humans, that have one baby, only has one mature egg at the time of ovulation, which is only one certain time at the month. So we cannot know when that time of the month is, and also we want to collect more than one egg from each female. And unfortunately, in rhinos, we cannot do the whole hormone treatment like in women, where you get to mature more eggs than one at a time and then aspirate them. So we have to now let this egg believe. Like, first of all, we put it in flushing media, we put it in holding media, get it to the lab, and then you have to make this egg believe that it's still in the ovary and that it wants to mature. So this is a process that takes about 2 days. After 2 days. So these are the freshly collected eggs. This was from one postmortem one, and so there was a good harvest there. And then after two days, if they're mature, they form a little polar body, and then we can inseminate them. So there's 2 ways of inseminate. The one is IVF, which means you just put the eggs with a certain amount of semen and let nature do its course. Or you do an ICSI. But with ICSI, this is long-term intracytoplasmatic sperm injection, just means you have this little device that sucks the egg and fixates it, and then you have a needle, and that is a little sperm in there. And then you inject it right into the egg so that it hopefully becomes an embryo. You see here, then it gets checked in certain time intervals, days, and then we see we got a 2 cells embryo there, and unfortunately we haven't been able to get to a blastocyst stage, which is sort of the stage that you need to be at to freeze them safely, because this is still very sensitive to freezing and also not really transferable. But there is another team in the world that already have produced embryos. So this is the one that I talked about earlier. This is what people have in mind when they think about embryo transfer, but this is actually what gets transferred. So in order to find the right time, you need to know the cycle of the female very well. And for that, we actually need trained females that we can daily monitor, either through blood sampling or repeated ultrasound, so that we find the time when she's in heat, and then we can determine when she would be ready to transfer a day 6 or day 7 embryo. And just to give you some ideas about where assisted reproduction has already played a role in meager herbivores like the rhino and the elephant. This is my colleague Imke. She actually started the Cryovault in 2018 with Hemmersbach. And then she is a reproductive wildlife specialist who works in zoos all over the world and does mainly reproductive work. And she has already successfully inseminated meager herbivores and created calves. And this one, for example, is a little calf born in, I think that was San Diego. And I can't highlight it enough, but collaboration is really the most important thing in all of this. What we do with the Cryovault has absolutely no commercial value. There's no, at no point of time are any of these samples going to be sold. This is purely to actually set up a biobank that's a backup plan for while we still have time to do so, and to utilize all the opportunities that are there. All of what we do gets scientifically written up and gets published in peer-reviewed journals, so that we share our knowledge with people, and we actually want to promote a broader use of it. And then also for certain research projects, this was my own, this was my PhD. That's how I started actually with rhinophores as a PhD student. Then at the moment I'm a post-doc student, and then from next year I'm going to be an extraordinary lecturer and have my own master's student, so it continues. And we just we just hope that we get to the successful calves at some stage. And I think I need to also highlight how important it is to get opportunities, because without opportunities here, we will not have any sampling. And I want to thank, especially in this area, Salati, who have been very, very supportive and forward-thinking. They have a good research team who understand the importance of research. And yeah, of course, nothing of this would be possible if it wasn't for people like Rhinophores or the CEOs of Rhinophores, who had a bit of an out-of-the-box thinking, but also actually fund all of this to be done now. Because if we look at the example of the northern white rhino, it's already too late. Like, we have 2 females left. We have some genetic backup, but it's not enough. And if you looked at our numbers, we have about almost 50 males. If you think when Ian Player brought the black rhino, the rhinos back, there was a population left of about 60 to 100 animals, and that was enough to recreate the southern white rhino. So actually what's in the biobank from the male point of view is already almost there. So we just need to react now before we lose more animals and we don't have those opportunities anymore. Yeah, and that's how I want to end it. Are there any questions? It's probably quite a lot to digest. So I hope it wasn't too scientific. It's difficult to break it down into understandable terms. Hi.
You mentioned you choose the male swimmers based on how straight they swim. Is there kind of any data to suggest that that's a good measure? Or?
Yeah, so it's actually one of the parameters that normally gets looked at. Progressive motility, it's called. So that is one of the main things you want to look at if you look at AI. So artificial insemination, because then you need sperm that knows where to go. If you use them for ICSI, where you just have to get one, it's not really that important as long as their DNA is correct and as long as they're alive. And their acrosome, so that's the tip of the little head, is intact and can fertilize. But for the rest, so that's one of the parameters that is actually in a normal semen collection, in a normal semen evaluation, is one of the parameters that gets looked at. Yeah?
Is there an ideal timeline from when you harvest to when the young is born? As in you've harvested the semen and done the artificial insemination. So when the calf is born, is there an ideal timeline? How long would that take?
So if you obviously, so you have to repeat the question. So the question is if there is an ideal timeline from the collection to when the calf is born. So that obviously depends on if you want to freeze it first or not. If you use fresh semen, if we would collect something now, we want to use it as quick as possible because every hour it loses quality. There's ways to preserve it, cool, ship it cool and that. But we preserve everything anyway, unless we do plant ICSIs like we've done where we collect the eggs, then we actually use a fresh sample. So that's as quick as possible. But once it's in liquid nitrogen, it doesn't actually matter because you've put it you kind of froze it in time. So if you take it out tomorrow or in 30 years, as long as it was maintained proper, should not make a difference. Yes?
The post-mortem retrieval success rate similar between males and females?
No, actually not. So the eggs are much more sensitive, and also you have to cut the whole body open. You have to obviously. Often the body already overheats much more. Sorry, I didn't repeat the question, Kai. Did everyone hear the question? Okay. If you the body actually overheats quicker, and because the testicles hang further outwards, it's actually more like a little bit safer. But yeah, it all depends. It all depends on the circumstances. But I would say that definitely the male is more successful. Yeah.
Do you know if black rhino and white rhino, can they breed? If they would?
No, they're two different species.
Totally different. No hybrids or anything.
Not by my knowledge.
So you couldn't put a black rhino embryo into a white rhino?
Well, see, they've done interspecies things. So the question was if you can breed black and white rhino, or if you can use actually, I think your question is if you can use maybe a white rhino as a surrogate. Yeah. So, Hamish, you might know, but they did this did they try to do buffaloes and cattle, but at some stage it always goes wrong. So even if they take, you unfortunately at some stage in the pregnancy there's just not enough, it's just not similar enough. To give you an example, from the semen that I collected, I talked about extenders with cryoprotectants, and we had two different extenders, and I did a study on which one works better. And one that worked very well in white rhino killed all the black rhino semen. So that's already how sensitive it is and how the species difference are there. Obviously, we try as much as possible to use a domestic animal as an idea, or as a background, as a model. But it can only go to a certain extent. So yeah, that's also the animal factor, or the limitation of animals. And the difficult part when you work with wildlife is they're intractable. You don't have that many. While if we test something in domestic animals, we can choose as many animals as we want. So that is one of the major. I think I didn't mention that, but that's one of the major obstacles. Any more questions? Yeah?
It's not a question, it's just a statement to say thank you for everything that you do.
Oh, thank you. It was just a thank you. Well, a nice thank you. Another question, yeah?
You had 100 attempts and about 77 success.
Yeah.
How much of that was post-mortem versus...
No, that was just the stats on live collection. So the question was if how many of the total numbers, how many were post-mortem, and how many were live collections. Actually, most of our collections are live collections because we don't make the time frame. Like often I collected it and I have the semen, but then it's already all dead. Yeah. But we work all over. So the black rhino, for example, the one population is in the Eastern Cape, and a lot of our work is in the Northern Cape because there's a rhino owner who collaborates very well. So we get opportunities there. He has some females that don't breed, and actually, funny enough, after we do the the egg aspiration, we found that they actually started breeding again, and it's actually well, they call it in science, because the other research team has also found this, they call it a reset or restart or something. So yeah, it has actually helped him. So now he's very keen to get the females that don't breed checked. Yeah.
One of your very earlier slides, you're showing something about de-extinction. And I was wondering, like, because I've been reading some stuff that people are trying to see if they can bring mammoths back because they've got DNA. Whether they could use elephants, not elephants, surrogates. Are they separate species? Was that even something possible?
So the idea of what they want to do, that's the next step. So this is manipulating their DNA as well. So the question was about bringing the mammoths back and if that's a possibility. So what you need to do is then you put that DNA into actually an egg or sperm of elephants, and then put it back into the elephant so that the elephant treats it as an elephant. But I don't know how possible that's going to be. That's on a whole nother level. But they're going to try.
Yeah.
Don't see any more questions.
Thanks, Janine. That was awesome. I think if we can give Janine one more round of applause for the fantastic work she's doing. Janine and Rhino, of course, I think super complicated stuff.
Yeah, it was
Yeah,
complicated.
very awesome to see. So thank you very
Thanks.
much. And thanks to all of you for coming out to another talk. As usual, I'm sure you know the rules by now, but if you can just head outside for a couple of minutes to set the staff and us reset the tables and everything. Please grab a drink, grab some food, enjoy yourselves, break some more glasses while you're at it. Come and ask Janine about 42,000 questions more. And just enjoy yourselves. And thanks everyone. We're probably selling shirts outside as well, so if you haven't got a shirt, go and grab one. Thanks everyone, have a good evening.